Hi, this is Dr. Swapna and I welcome you to your Learn Lab. If you are an IGNOU student looking for help in your studies, then you have reached the right place. Here we have lecture videos on different core and elective subjects of important IGNOU courses. We aim to help you ace your exams. So hit subscribe to our channel, and as you watch, make sure to like and share too. Come and join me on this beautiful learning journey. In this video, we are going to study about hemispheric specialization, which is a part of block 3 titled brain lateralization. It's a very small block or a very small unit. And in this we are going to learn what is left and right hemisphere. We have already studied about it before, that there is a central fissure that divides our brain into two, left and right hemisphere. And these two hemispheres are connected by what? Corpus callosum. And we'll see what is a split brain condition, where if this corpus callosum is cut, how the two halves behave. Then we'll also see methods to study brain lateralization. Okay. First, before we delve into what is brain lateralization, let's look at the left and right hemispheres of the brain. Our brain is divided into two halves, right hemisphere and left hemisphere. They have exactly the same structure. Same cranium covering it, same meningeal layers, same cerebrospinal fluid, same cortex, midbrain and hindbrain, right? They are separate from each other, but they do exchange information through a number of channels. So even though they are separate, they have modes or channels for exchanging information. And which are those channels? The two halves are connected to each other by different bundles of nerve fibers. Corpus callosum, we have learnt about this, right? This is the band of nerve fibers which are connecting left and right hemispheres. Other than corpus callosum, we also have anterior commissure, hippocampal commissure, massa commissure, posterior commissures. Commissure simply means bands of neurons. Okay, and these are all just locations: anterior, hippocampal, massa, and posterior. Other than these commissures, we also have optic chiasm. This is the part of the brain where the optic nerves from each eye cross over, forming a X shape. So we have two eyes. Nerves that are arising from both these eyes crisscross at some point. Okay, this area is called optic chiasm. Alright, so I'll show you these parts. See, look here. We have the corpus callosum here, which is the most prominent band which separates the left and right hemisphere. Then we have anterior commissure here. We have optic chiasm here just behind your eyes. Then we have the posterior commissure here. Anterior means in the front, posterior means in the back. So we have an anterior commissure, then we have a posterior commissure. Besides that, we have a hippocampal commissure. Where is it? At the hippocampus. Then we have a massa commissure here. Alright, so these are the different bands of neurons which are connecting the two hemispheres. The two halves are contralateral in function. This means the left hemisphere is connected to the right side of the body and right hemisphere is connected to the left side of the body. Okay, so now do you understand why optic chiasm is important? The two eyes, nerves coming from two eyes are crisscrossing. Why? Because the nerve coming from the left eye has to go to the right brain, and the nerves coming in from the right eye has to go to the left brain. So this crisscrossing happens at the optic chiasm. So that's why this is important. So the two halves are contralateral in function. The two halves also exhibit hemispheric specialization or lateralization. This means each hemisphere specializes in certain functions. The function that left hemisphere is specializing in may not be the dominant function for the right hemisphere, and vice versa. But both the hemispheres coordinate together, resulting in the functions of the brain. So you understand what is hemispheric specialization now? Each of the hemispheres specializes in certain functions, and both the hemispheres coordinate together, resulting in the complete brain function. Alright. Now, have you ever wondered why most people write with their right hand? Or why a brain injury in one area can suddenly make someone lose their ability to speak, but they can still understand what you're saying, right? So this came from the idea of cerebral dominance. The idea of cerebral dominance was popularized by a neurologist named John Hewlings Jackson in late 1800. But the work on it progressed in early 20th century thanks to the researchers like Paul Broca, Carl Wernicke, and later on Roger Sperry. So you remember Broca's area and Wernicke's area? We talked about this when we were studying cerebral cortex. No? You can go back and watch that video, right? I'll put the link in the description. So uh, I'll tell you the story, okay? How Paul Broca came to discover this part of the brain, which today is famously known as Broca's area. So, back during Broca's time, a man named Louis Le Borgne was admitted to a hospital in France. And it's funny why he was admitted, okay? Because for years the only word he could say was tan. He understood everything he was told. He could answer anybody using gestures, but he was not able to say anything back other than tan. He was mentally present also, but no matter what he tried to say, when he opens his mouth, the only sound that comes out is tan. And because of this, he also got a nickname. You can guess it. Yes, tan. So when this person died, Broca performed a post-mortem autopsy on this person, and he discovered that a specific area in the left frontal lobe of his brain was damaged. So he understood that it's because of this damage to this area that Lewis could not talk. All he could say was tan. And because of this, this area is now famously known as Broca's area and is responsible for the speech production. And we know that although both sides of the brain look similar, certain functions like language are dominated by one hemisphere, like how Broca showed that left frontal lobe, right? So in most people, the language dominance is the left hemisphere. Now, this was the birth of the story of cerebral dominance. So, the theory of cerebral dominance, it states that one hemisphere of the brain, usually the left, is dominant for specific cognitive functions, especially language and fine motor control. Okay. The non-dominant or the minor hemisphere, which is usually the right hemisphere, supports functions like spatial awareness, musical ability, and understanding emotional tones. Okay, so this was theory of cerebral dominance. It says that the left hemisphere is the dominant hemisphere and it is responsible for cognitive functions like language and fine motor control or control of the movement. And right hemisphere is the non-dominant or the minor hemisphere which supports creative functions like musical ability, understanding emotions and spatial awareness. Alright, but later on when we had brain imagings, neuroimagings, all those latest techniques, we know that this is a myth. The theory of one hemisphere being dominant is inaccurate because both sides of the brain work collaboratively and individuals don't fit neatly into left brain or right brain regions. 90 to 95% of people who are right-handed have dominant left hemisphere. But in left-handed people, this pattern is not predictable too. They could be either dominant left hemisphere people, they could be dominant right hemisphere people, they could also have both hemisphere equally dominant. Okay, so even though we call left brain people logical and right brain people creative, the reality is much more complicated than that. We know that both the hemispheres work in coordination with each other. Alright, so that was theory of cerebral dominance. Let's now look at some of the functions in which the left brain dominates in and some of the functions where the right brain dominates. First, we have language dominance. Left hemisphere is involved in spoken and written language, including comprehension and production. Cerebral lateralization was discovered while language abilities were being studied. If you remember, the Broca's area, which is responsible for speech production, and Wernicke's area, which is responsible for speech comprehension. Both of these parts are present in the left hemisphere or the left part of the brain. Alright. Next we have ipsilateral movement. Ipsilateral movement means same side. Movement controlled by the same side of the brain. It's opposite to contralateral, which means that the left brain controls the right part of the body and the right brain controls the left part of the body. This is contralateral. Ipsilateral would be right controlling right and left controlling left. Alright. So left hemisphere shows more ipsilateral control of motor activity, particularly for complex movement, which means left brain has more control over left part of the body compared to the control right brain has over the right part of the body. I hope that was easy to understand. Left has more control over the left part of the body than right has over the right part of the body. This is ipsilateral and not contralateral that we are talking about. Next we have spatial ability. Right brain is superior to left brain in spatial abilities such as direction, distance, geometry and depth perception. It also helps in navigation and object recognition. This means if someone has a dominant right brain, they'll be really good at map reading and navigation. Then we have emotional stability. So the right brain handles emotional responses and intuitive understanding. Damage in right brain prevents an understanding of emotional responses such as humor and sarcasm. So if someone is not able to get any humor or a sarcasm, which means they don't have a dominant right brain. Alright. Then we have musical ability. Kimura used dichotic listening to prove that right brain is superior in musical ability. Now, do you remember what is dichotic listening? Dichotic listening was the testing method where a person is given two different sound stimulus from each of his ears. Like the right ear will have one kind of sound coming in and the left ear will have a different kind of sound coming in. And then the patient is asked specific questions about each of those which the person has to respond to. Alright, so that is dichotic listening test. And Kimura used dichotic listening to prove that right brain is superior in musical ability. Right hemisphere lesion disrupts musical abilities in patients. Then we have differences in memory. Both hemispheres are involved in creating and storing memory, but left hemisphere is involved in verbal memory while right hemisphere is involved in non-verbal memory. Anything that is related to words, the left hemisphere remembers it. If it is pictures or sounds, which is something that's not related to words, then the right memory will be remembering it. Here I have a table. This one is present in your study material, so I'm including it here. So they have given 7 categories of functions and the role of left hemisphere and right hemisphere. In case of vision, if someone has left dominant hemisphere, then they would be good at words and letters. If the person has a dominant right hemisphere, then the person would be good at faces, geometric patterns and emotional expression. Audition, which means sound. Left hemisphere dominance means they'll be good at language sounds. And right hemisphere dominance means non-language sounds like music. When it comes to touch, left hemisphere is not recognized as of now. But right hemisphere is good at tactile patterns and braille. Braille is the language used by blind, right? They touch and understand the letters. So right hemisphere is dominant in case of blind patients who are able to use braille. About movement, left hemisphere is dominant when it comes to complex movement and ipsilateral movement, which means same side. And for right hemisphere is good at movement and spatial patterns. About memory, we learned that both hemispheres are good at memory, but left hemisphere dominates in the field of verbal memory, finding meaning in memories, while right hemisphere is good at non-verbal memory, perceptual aspects of memory. Okay, then language. Left hemisphere is concerned with speech, reading, writing and arithmetic. Right dominant hemisphere, emotional content. Spatial ability, again, it's all right hemisphere. Mental rotation of shapes, geometry, direction and distance. So these are some of the functions where it is believed that left hemisphere is dominant and where right hemisphere is dominant. But we know that both the hemispheres coordinate with each other to bring out the functions of the brain. Now let's study about split brain. Let's see what is a split brain and how it affects brain lateralization. Split brain refers to a condition where corpus callosum, the main pathway connecting the two hemispheres of the brain, is severed or damaged. So, if the corpus callosum is cut, the two hemispheres will be functioning independently of each other as individual brains. So, this condition is called split brain condition. Meyer and Sperry, they are the pioneers in studying split brain condition. They perform different experiments on different animals to see what happens when corpus callosum is severed. Okay, so we look at one of the experiment conducted by Meyer and Sperry on cats. So in their experimental setup, they had three control groups. Now the control group means these are the group which do not go through the experimental condition, or whatever the experimental condition is, it is not applied to the control group. There would be a separate experimental group on which the experimental conditions will be applied or the experimental treatments will be applied. Control group will be kept as it is, not affected, not treated or not given the experimental condition. So the first control group were cats with intact brain. The second control group was cats with severed corpus callosum. The third control group was cats with severed optic chiasm. We remember what is the optic chiasm, right? It's that part where the nerves coming in from left and right eye cross each other, right? So these were the three control groups. Then the experimental group. Cats with both corpus callosum and optic chiasm severed. Now, this fourth group, which had no corpus callosum and no optic chiasm, they had two totally independent hemispheres, and that's why they were used as the experimental group. What was the principle they were using in their experiment? They were using the fact that corpus callosum and optic chiasm are the only two routes through which visual information can travel from one hemisphere to the other. We have a lot of commissures crisscrossing we have learnt, but here what they are concerned is about visual information, and their principle says corpus callosum and optic chiasm are the only two routes through which visual information can travel from one hemisphere to the other. Let's look at the actual experimental procedure now. They performed the experiment in two phases. In the first phase, the cats were trained on a liver press pattern discrimination task with a patch over one eye, which means one of the eyes was covered using a patch, and then the cats were taught to, press a lever. There was an experimental setup where there were two levers. Now pressing one of the lever, the cat would receive a reward, which might be a food, or maybe the gates open and they are free to go out. So using that one patch, the cats were trained on liver press pattern discrimination. So what happened? The result, all the groups, including those key experimental groups, could learn the task easily. We had three control groups, one with intact brain, one with severed corpus callosum, one with severed optic chiasm, and the experimental group with both corpus callosum and optic chiasm severed. So the result was all the four groups could learn the task easily. They all learned that pressing which of the lever gives them the reward. Okay. Now what was the inference from it? The inference was that a single hemisphere can learn any task independently as efficiently as two hemispheres together. So the cats need not have completely connected hemispheres. Even if the cat has a connected hemisphere as the first control group, and even if the cat has a severed corpus callosum only, or the cat has severed optic chiasm only, or the cats have neither corpus callosum nor optic chiasm. In all of these cases, the cats could learn the task independently and as efficiently as each other. Now, in the second phase of the same experiment, the patch was switched to the other eye. So, for example, in the first phase, if the patch was on the right eye, in the second phase, the patch was shifted to the left eye. And the cats were trained to do the same exercise, the liver press mechanism, right? And what was the results? Cat with only optic chiasm severed or the corpus callosum severed maintained their performance. Okay, this means they had either optic chiasm present or they had either corpus callosum present. In both of these cases, one of the connections for transferring or transmitting visual signals were present. So the performance was maintained. However, the group with both the corpus callosum and optic chiasm severed struggled, requiring relearning as if the task was new. What happened here is that the three control groups, first with the intact brain, those cats could learn the task easily the second time because they have already studied it the first time, they know what exactly it is, so their performance continued. Same with the second category, which had the corpus callosum severed. Why? Because they had intact optical chiasm, because of which whatever their left eye learnt was transmitted to the right eye also. The same thing happened with the cats which had only optical chiasm severed, because whatever their left eye studied or learnt about the liver was transferred to the right eye also through the corpus callosum. Now the last group, the experimental group, which had both corpus callosum and optic chiasm severed. There was no means for the learning to be transferred to the other half, because of which when the patches were removed and shifted to the next eye, the cats had to start learning the whole thing as if they are learning the first time. Their right brain did not know what the left brain already had studied. Okay, so this was the result of the second phase. And what did they infer from here? That hemisphere can act as separate brains. Corpus callosum transfers information across the hemispheres. I hope you understood that. That was easy to understand, right? Let's see what was the outcome of the experiments Meyer and Sperry conducted on the cats. The brain hemispheres can function as independent units capable of learning and storing memories. This they showed from changing the patches on both the eyes of the experimental group of cats which had both corpus callosum and optic chiasm severed. So both the hemisphere in that cases learned the task independently as if they were learning it for the first time, which means the brain hemispheres can function as independent units capable of learning and storing memories. The corpus callosum and optic chiasm facilitates interhemispheric communication and information transfer. The best strategy for studying corpus callosum function is to limit information to a single hemisphere, right? So if you limit the information only to a single hemisphere by closing one eye or closing one ear, you will be able to limit that information from passing on to the next hemisphere, because there is no connection between the left and right hemisphere in split brain patients, right? So now after studying split brain in cats, Sperry moved on to studying split brain in human patients. The split brain condition was induced in humans by conducting commissurotomy, a treatment for epilepsy by Vogel and Bogen. Now, we have learned about commissures. These are the tracts of neurons which connect left hemisphere with the right hemisphere of the brain. We also know what epilepsy is. This happens when there is a surge of electrical activity in different areas of the brain. So, to treat epilepsy, this surgery is done in where these commissures are cut off so that electrical activity from one hemisphere is not passed on to the second hemisphere. Now, these surgeries were life-changing for patients suffering from intense seizures, but they also created a unique opportunity for scientists to study how two hemispheres function independently. And this chance was used by Sperry and Gazzaniga. They conducted experiments on such patients. Their methodology involved limiting information to one hemisphere and studying the effect of it. Alright, so in this way, they didn't have to breach any ethical codes because conducting such experiments on humans, it evokes a lot of ethical concerns and questions. But here, the patient had already got the surgery just for curing or controlling seizures, and so they could easily conduct their experiments on them. Let's see what the experiment was. In their experiment, Sperry and Gazzaniga presented visual and tactile stimuli. Tactile means touch to each hemisphere independently. How did they do that? Let's see how they presented the visual stimuli. Okay. The patients were made to sit facing a screen or a display board. A small dot was placed in the center of the screen, and the patients were instructed to focus their gaze just on this dot throughout the experiment and nowhere else. Now what they did is that they showed visual stimuli like word or a picture or an object for very short duration of time. Short, when I say, it's like 1/10th of a second or 1/20th of a second. You know how short 1 second is, right? So imagine 1/10th and 1/20th of a second. And also they flashed it to either the left side of the dot or the right side of the dot. Why did they do that? So that this brief exposure ensured that the stimulus was only processed by the contralateral hemisphere. Okay, what does meant by contralateral hemisphere? That whatever is flashed on the left side of the dot will be seen by the left eye and processed by the right hemisphere. And whatever is flashed on the right side of the dot will be seen by the right eye and processed by the left hemisphere. And this would be so quick so that the eyes will not be able to move or the other hemisphere will not be able to find about it. Which means when the object was flashed on the left side of the dot, it was so quick that the right eye did not have the time to look at it, turn the face or turn the head or look at it. So this way the information will be restricted to only one eye and one hemisphere of the brain. Okay, so by presenting images or words to the left or right of the visual field, the researcher could isolate the information to the right hemisphere or the left hemisphere respectively. Now, about tactile or the touch stimulus. So, for this what they did is, the objects were placed on the patient's left or right hand without them seeing it, like maybe under a table. Now, because each hand is controlled by the opposite hemisphere, which means right hand is controlled by the left hemisphere and left hand is controlled by the right hemisphere. This tested whether the hemisphere controlling that hand could identify the object or not. So, objects were placed in the patient's left or right hand, allowing for assessment of the right or left hemisphere's ability to process the tactile function. So in some cases, they were also asked to match objects or draw what they saw. Match objects in the sense, they'll be shown the picture and they'll be asked to pick that object from a group of objects kept under a table. They just have to touch, identify and find it out. Okay. And they had to do this using either the left hand or the right hand, not the both hands. Okay, so this was how they conducted their experiments on split brain patients by presenting them visual and tactile stimuli to only one of the hemispheres. Let's see what they observed from their studies. Patients were observed while performing various tasks such as describing what they saw, drawing or pointing, and matching objects. So they observed that when a stimulus was presented to the right visual field or right side of the dot, which is controlled by the left hemisphere, like whatever they see through the right eye is controlled by the left hemisphere. The patients could easily verbalize and name the stimulus easily. They could use words to tell exactly what they saw. Now this was because left hemisphere typically controls the language that we have already studied. When the stimuli was presented to the left visual field or left side of the dot and seen by the left eye, which is controlled by the right hemisphere, the patients were not able to tell what they saw, but they could easily touch and identify or draw what they saw with their left hand. Okay. The right hemisphere lacks language ability and hence could process images, spatial info and emotions, but it could not produce speech, while the left hemisphere, which controls language, could produce speech, right? So the right hemisphere could not verbalize what it saw on the left visual field. The right hemisphere could draw or point to objects seen in the left visual field using the left hand. The patients could match objects placed in the left hand even though they couldn't verbally name them. Alright, so this was what they observed from their experiments. Now let's see what were the outcome of the experiments conducted by Sperry. First one, hemispheric lateralization. So these experiments revealed that the left hemisphere is dominant for language and verbal processing, while the right hemisphere is more involved in spatio-visual tasks and non-verbal communication. Okay. Then independent function. The hemispheres could function independently, but communication between them was severed or disrupted after corpus callosotomy or severing of the corpus callosum. Left hemisphere dominance. The left hemisphere could name objects presented in the right visual field but not about those in the left visual field. Similarly, the right hemisphere excelled in visual motor tasks such as drawing or picking up objects presented in the left visual field using the left hand. While Sperry was busy studying split brain, one of his team members called Zadel developed something called Z-lens. And why? To study the functional differences between two brain hemispheres in individuals who had undergone a split brain procedure. And Z-lens helped Sperry a lot to study split brain in humans. What is Z-lens? It is a specialized contact lens that allows for the controlled presentation of visual stimuli to one hemisphere of the brain at a time. The Z lens is opaque on either the left or right side, so that when a person is using the Z lens, they can see only through one eye, either the left or the right. Okay. It is designed to block visual input from one side of the eye, effectively isolating visual information to the corresponding brain hemisphere, which means if the left is blocked, only the right eye will see, which will be processed by the left brain. Similarly, if right is blocked, only left will see, which will be processed by the right brain. So this was the function of the Z lens. By using the Z lens, researchers could demonstrate that each hemisphere of the split brain has its own separate visual field with distinct processing abilities. For example, a visual stimulus presented to the left eye projected to the right hemisphere of the brain would be processed and understood by the right hemisphere only. Same stimulus presented to the right eye projected to the left hemisphere would be processed by the left hemisphere only. So that was the use of Z lens, which Sperry used in his split brain studies. So this was all about split brain and studies on split brain, which told us the specialization or dominant functions of each hemispheres of the brain. Let's see what are the other methods to study brain lateralization. First, we have sodium amytal test. It's also known as VADA test after the neurologist June VADA. It's also called intracarotid sodium amobarbital procedure or ISAP. Intra means inside. Carotid, it's a name of one of our major arteries. Okay. Sodium amobarbital is the chemical. It's a diagnostic tool used in split brain studies and epilepsy surgery planning to determine which hemisphere is dominant for language and memory. Okay. It involves temporarily anesthetizing one hemisphere by injecting sodium amobarbital into the internal carotid artery, and it allows researchers to assess the function of the remaining hemisphere. So what happens here is that, so one side or one hemisphere is anesthetized by using sodium amobarbital. So in this way, no function happens in that hemisphere. The other hemisphere will be the only functional part of the brain. And then the researchers see what all functions the patient is able to perform without any difficulty, and what all functions the patient is not able to perform. So those functions that the patient is not able to perform will be attributed to the part of the brain which is non-functional due to anesthetizing by sodium amobarbital. Alright. The second technique we have is dichotic listening test. We have already studied about like dichotic listening, right? It's a psychological experiment where different auditory stimuli are presented simultaneously to both ears, allowing researchers to study selective attention, brain lateralization, and auditory processing. The participant is presented with two different auditory stimuli through earphones. So, the left ear will be presented with one kind of auditory stimuli, and the right ear will be presented with another auditory stimuli. And then the participants are asked to focus on one ear or both and then report what they heard. Researchers analyze the accuracy of the reports and the frequency of responses from each ear to understand how the brain is processing the information. So they'll ask questions regarding the stimuli that was presented to right ear and the left ear, and then they check how accurate the answers are, and based on that they understand the processing of the brain. Here, another scientist called Kimura, he observed that most patients could reproduce the information presented to the right ear than the left ear. We know that right ear is controlled by the left brain, right? And the left ear controlled by the right brain. He saw that the information that was presented to the right ear was correctly reproduced by the patients. This showed that left brain was responsible for language and verbal memory. Alright. Next we have functional brain imaging. We have studied neuroimaging in methods to study biopsychology, right? So this is a part of it. Here what happens? A volunteer is engaged in an activity such as reading or playing games. And when the person is engaged in these activities, the activity of the brain is monitored using PET scan or fMRI. What is PET scan? Positron emission tomography. What is fMRI? Functional magnetic resonance imaging. These are non-invasive techniques, which means the studies are conducted from outside the brain. There is no surgery involved, right? fMRI relies on the principle that when a brain region is active, blood flow to that region increases, bringing more oxygen-rich blood. This we have studied in detail in our video on methods to study biopsychology, neuroimaging techniques, right? So what does fMRI do? The machine detects these changes in blood flow and oxygen levels by using a strong magnetic field and radio frequency pulses. These changes in blood flow and oxygen levels are used to create images that show which part of the brain are active during the task that is being performed. Okay, so that was all about methods to study brain lateralization. And with that we come to the end of unit 5 block 3. Okay, so caution, yeah, people do say that left brain is for logic and right brain is for creativity, but it totally depends on which part of your brain is functional and dominant. Okay, both part of the brain are equally required and they coordinate and work together to affect the complete brain function. It's not just one hemisphere. You need both of your hemisphere working together in tandem for you to function properly. Alright, so next we look at previous year questions from this part. So every year question comes from this topic. Sometimes even two questions from the same part. This is a very small block. The only topic here in this block is cerebral lateralization, which we have completed in this video. Yet you will see the number of questions every year it comes, and sometimes more than one. June 2024, the question came, describe cerebral lateralization of functions with examples. So you have to write about cerebral lateralization here. December 2023, how does severing corpus callosum impact behavior? You have to write about Sperry and Gazzaniga, the split brain thing, and what happened when corpus callosum was cut. Okay. Also, the same year, same question paper, there was a second question. Define hemispheric lateralization. Discuss the methods to study brain lateralization. Okay, you have to write what is hemispheric lateralization. You also have to discuss the three methods that we have studied. Then June 2023, discuss the methods to study brain lateralization. December 2022, explain Myers and Sperry's experiment on the split brain. So if they ask Myers and Sperry, it's the cat experiment, okay? If it is Myers and Gazzaniga, then it's the human split brain experiment. Then June 2022, explain the cerebral lateralization of functions. It doesn't end here. We have December 2021, where this part question came. Write short notes on the following in about 150 words each. Split brain studies, methods to study brain lateralization. Then December 2020, bring out the differences between left and right hemispheres. We have the table. You just learn that and explain it a bit. Then the same year, 2020 December, we had second question, which was explain the effect of split brain in humans, where you have to write about the Sperry and Gazzaniga's experiment. What was the observation? What was the outcome? Alright. So that was all about brain lateralization or hemispheric specialization. Right? Thank you all for learning with me. If you found the video helpful, make sure you let me know by hitting the like button. Make sure you share the video with your friends too. Subscribe to make sure that you don't miss out on any of the new videos. You can reach me by simply dropping a comment. See you in the next one. Till then, happy learning.